Again, this time, let me start by saying it's a real pleasure to be here in São Paulo and in Brazil again. I love to visit, and it's very exciting for me to come. So this time I will be talking about sort of the larger picture and maybe a more forward-looking picture about breast tumor subtypes, with an emphasis on the triple negative subtype. I could give a 1-hour talk about HER2 positives, ER positives, triple negatives, so this time I will talk about triple negatives. In large part because if we made triple negative its own group, it would be tied for 5th place in terms of cancer killers of women in the United States and probably in this country as well. So it by itself is really a significant disease group. And actually, we had some recent papers showing we should really consider it its own disease. It's in some ways completely different than ER positive breast cancers. And we should really, again, think of it as another type of disease that occurs in the breast. Of course, over the years, we've come to describe triple negative cancers using gene expression patterns. But as we all know, triple negative breast cancers is a very biologically heterogeneous group. And so I'll first tell you a bit about that biological heterogeneity, and then focus on some of the genetics, how we can therapeutically target those cancer-driving events, and then actually I'll end with some thoughts on immune therapy. So in this review, Elise Pratt and myself took a set of 1,000 patients, and actually we broke them into groups according to clinical status. So we asked, if we take the triple negatives and then look at what are their intrinsic subtypes, what are the results? We kind of looked at it the other way as well, saying, I'm going to look at basal-like breast cancers and then look at what the clinical features of basal-like breast cancers are, right? So if we take triple negative breast cancers and look at the different intrinsic subtypes within them, about 80% of triple negatives have the basal-like phenotype, and then there's almost an equal representation of all the other subtypes. If we start with basal-like group first, and then ask what's the clinical breakdown of the basal-like group, about 70% of basal-like cancers are triple negative, but that also means 30% of basal-likes are not triple negative, right? And those patients end up in our HER2 studies. They end up in our endocrine therapy studies. And in some ways, they're treated just like an ER positive patient, ignoring the fact that it has this very different genetic background. And I think we should not ignore that fact. So, many of you are also aware of our work on the Claudin-Low type, which has stem cell-like features. If we include it within the triple negative spectrum, it is a major part, potentially 20%, but nonetheless, the majority, the overwhelming majority of triple negatives are still of the basal-like phenotype. So I believe that if we are to make therapeutic progress against triple negative breast cancer, we need to focus on the basal-like group because it is the biological, it is the most numerous group, it is a relatively homogeneous biologic entity, and I think we need to be thinking in that way. Certainly, there are many interesting and promising studies occurring against the androgen receptor within triple negatives that predominantly identifies these luminal cancers, luminal A, B, HER2 enriched, that are present within triple negatives. However, I actually think we're still missing the big picture here, right? We need to look and focus on the 80% that are basal. And many studies have literally forgotten these. So, as I'll say, I'm now going to spend the rest of the time talking about the basal-like subtype. What do we know about the genetics, and what can we do about it in the clinic? Right? So, first thing we know is that if left untreated, these patients have a poor prognosis. So we have to do some treatment for them. We called them basal-like because they stain positive for keratins like keratin 5 and 6 that stain the basal layer of your skin or the basal layer of your airway epithelia. And in other publications, we've actually shown they have genetic and expression features similar to lung squamous-like cancers as well. So I can argue that maybe we only discovered basal-like cancers probably, 10, 12 years ago, so we had a lot to learn. One thing we've learned over and over and over again is a very strong association with p53 mutation status. And actually, in this recent, well, I guess this publication from 2012 from the Cancer Genome Atlas Network, we can see about 80% of basals have a p53 mutation. That's also shown here in this representation where each of these rows signifies a different subtype, and these little marks identify the mutations. You can see here basal-like cancers, 80% have p53 mutations, and for luminal A cancers, only 12% have p53 mutations. The other interesting feature is the type of p53 mutation also varies by your subtype, where the basal-like subtype tends to have nonsense and frame shift mutations, right? Suggesting it is definitely loss of function of p53 that is occurring in most basal-like cancers. So this 80% comes from using a single assay of exome sequencing, right? So it's probably even an underrepresentation of the actual p53 loss rate. So I'm going to say it's a required event in a basal-like cancer to have p53 loss. Of course, with all these gene sequencing efforts going on, certainly in the Cancer Genome Atlas, one thing you get is a list of somatically mutated genes that are common to all breast cancers, what we're looking at in the first column here, or I've highlighted in red those mutations occurring in basal-like cancers. And somewhat of the surprising answer is there are not very many recurrently mutated genes in basal-like breast cancers. There's maybe, statistically speaking, 3. p53, PI3 kinase, and retinoblastoma. And the other sort of dramatic feature is that we have p53 mutated at this 80% frequency. The next one, PI3 kinase, is at about a 10% frequency. So there's this huge jump from number 1 to number 2 on the list. So when we saw this, I literally scratched my head and said, well, what is then responsible for the aggressive behavior of basal-like cancers if there aren't many somatically mutated genes? And the answer is in the DNA copy number changes that are present. So in this publication, we looked at the common DNA copy number changes that occur in each intrinsic subtype. And what you can see here is that, for example, the Claudin-Low group had the fewest changes, and the basal-like group had the most. And these regions in black were actually regions that were either specifically lost or gained in one particular subtype. And once again, you can see the basal-likes have the most regions of black. So here, let me blow it up so you can see it. So here's a copy number gain on chromosome 1. Here are chromosome losses on chromosome 4q and 5q. Here's MYC. So now that we have these frequent events, on the DNA copy number landscape, now we can start to try and put names to what's being selected for in each case. And actually, what really sparked our interest are the ones I have in purple here, loss of chromosome 4 and loss of chromosome 5, which, along with gain of chromosome 8 with MYC and gain of chromosome 1, are the most frequent genetic changes in basal-like cancers, right? So what is happening here? Well, there are two genes in this region of interest, right? One, which I had never heard of until I saw this paper, is called INPP4B. It is functionally similar to PTEN in that it is a negative regulator of PI3 kinase signaling. So if you lose it, you activate PI3 kinase signaling. And actually what we can see is that in human basal-like cancers, INPP4B is at its lowest level, both on the RNA and on the protein level. And actually, interestingly enough, in our mouse models of basal-like cancer, INPP4B is also lost on the DNA level and low on the RNA and protein level. So the PI3 kinase pathway is a very important pathway for a number of reasons. First, because it's a cancer-causing pathway. It's activated in a large number of cancers, including breast cancer, and in theory, it's therapeutically targetable. There are many inhibitors being developed against PI3 kinase, against mTOR, against AKT, which all in some way, shape, or form are part of this pathway. If we now go look at the copy number changes in basal breast cancer, it pretty much targets every part of the pathway. You see common amplifications of AKT3, and you actually see amplifications of PI3 kinase itself. And you see loss of 3 different negative regulators of the pathway, right? So this pathway is probably activated in every basal-like cancer, not by mutation, but by copy number gain of the activators of the pathway or loss of the negative regulators of the pathway. And this was nicely summarized in one of our TCGA papers here, where here we're looking at protein or gene signatures of PI3 kinase pathway activity where yellow is high. And what you can see is that, it's a little bit blocked here, the basal-like group is right here, and they're all high for the pathway activity. Luminal A's actually all look to be low for pathway activity, which is a little hard to understand since luminal A's have a very high frequency of PI3 kinase mutations, about 40%. So somewhat of a disconnect there, but nonetheless, it suggests that PI3 kinase pathway is important in basal-like cancers. We can look at some of these other copy number changes, MYC amplification, RB loss, BRCA1 and P53 loss, right? So I can argue we know the genetic drivers of basal-like breast cancer. The answer is most of the time, there's not a lot we can do about it therapeutically, but hopefully that will change. One of the other things we know is associated with basal-like cancers is the BRCA1 pathway. And the first evidence for that came through this study where we reanalyzed data coming from the Netherlands Cancer Institute where they had 18 BRCA1 mutation positive patients done by gene expression profiling, and I believe it was 17 of the 18 had the basal-like phenotype. This has subsequently been validated in many other studies. So for reasons that we still don't fully understand, if you have a BRCA1 mutation, not only are you predisposed to breast and ovarian cancer, but you're predisposed basically to one type of breast cancer, and it's the basal-like type. So this link to BRCA1 status also brings in possible therapeutic links to sensitivity to PARP inhibitors and sensitivity to DNA damaging agents like the platins, including cisplatin and carboplatin. I'll talk about that a little bit in this talk, and I'll talk about it more in the talk I will give tomorrow. So what other genetic features did we learn about basal-like breast cancers in the TCGA? Well, we reconfirmed the association with BRCA1, and we reconfirmed an association with loss of RB. So that's good. And here just shows involvement of the RB pathway in this complicated figure from our TCGA paper. So in this case, blue over here means loss of RB function, right? And the basal-likes are homogeneously lost RB function, lowest for RB on the mRNA level. But again, it's not somatic mutation of RB. It tends to be the genomic deletion of that loci and potentially loss of other regulators of the pathway or amplification of cyclin E1. So again, I can argue we know the genetic causes of basal-like breast cancer: P53 mutation, BRCA pathway dysfunction, RB loss, PI3 kinase active. Now, in the clinic, what can we do about that, right? So of course, these are chemotherapy-sensitive patients. Again, I'll show you some data about that today and tomorrow. I'll touch on whether there's a benefit by carboplatin. I've made an argument to you that we should be looking at PI3 kinase pathways inhibitors, maybe also MEK pathway inhibitors. But I think in some ways the most promising is some of the immune therapy targets. And in part because it's certainly been known for many, many years that there are many breast cancers that have tumor-infiltrating lymphocytes, right? And in this case, here we have a breast cancer stained for antibody, showing that there are many B cells producing antibodies in a tumor. And actually, in probably the very first paper we had on microarrays, when we looked at breast cancers, we could see by gene expression patterns, immune cells are present, right? Antibody only comes from B cells, right? CD8, CD4 proteins only come from T cells. CD68 is a macrophage marker. So by gene expression, we can actually quantitatively see the presence of different types of immune cells and maybe even see what type of immune cell it is. So in a more recent publication using RNA sequencing data, we re-identified these immune cell signatures. And what you can see here is that the basal-like group, on average, tends to have the highest expression of these immune cell signatures. And at least half of basal-like tumors have many TILs in them, or however you want to measure immune cells, by gene expression, by pathology, by FACS sorting. It is really the basal-like tumors, the claudin-low tumors, and a subset of the HER2 positives that have these immune infiltrates. So in this publication, we showed a result that many others had shown, which is that within breast cancers as a whole, the presence of immune cells is a good prognostic feature. Actually, we kind of took it a step farther here, and it's a little hard to see, but actually, if you break down by subtype, it's only within the HER2-enriched subtype and the basal-like subtype where immune cells are prognostic, right? So in LuMa breast cancers, the large majority of them don't even have immune cells. And those that do, it doesn't make a difference in terms of prognosis. But if you're a basal-like cancer without immune cells, you're likely to do worse than a basal cancer who has immune cells. You can even do this looking at just expression of antibody variable region genes, right? So any good marker of immune cells will work. Actually, let me also point out one thing here, which is all these different immune signatures are highly correlated with each other, right? So if a tumor has B cells, it tends to have T cells and it tends to have macrophages. There's a fairly rare tumor where it has one but not the others. So I think that's important to keep in mind as we think about potential biomarkers for immune checkpoint inhibitors. Shireen Loy and others have also shown within triple-negative breast cancers, again, years before we did, right, that the presence of TILs is a good prognostic feature. So getting back to my earlier talk, in some ways this is level 1 evidence, right? Multiple studies all have shown the same feature, that if you have a triple negative, if you have immune cells, that's a better prognostic feature. Now, what that means in the clinic is to be determined, right? We definitely should not use this to determine who gets chemotherapy or not. These are triple negatives, they should probably all get chemotherapy. And how we measure immune cells is also a difficult thing. If we just look at TILs, the kappa score is not very good. And so we either need to improve the scoring of TILs or potentially use some of these gene expression markers. So I will now first introduce you to this CALGB study looking at the potential benefit of carboplatin and bevacizumab within triple-negative breast cancer patients. This study has been completed and reported in the JCO, and many of you probably know the results. It's maybe a little bit hard to see here, but here's the control arm. So when given ACT, neoadjuvant chemotherapy, the PCR rate in this population was 42%. If you add bevacizumab, it went up to 50%. If you added carboplatin, it went up to 50, I think it's 53%. If you add the two together, we had a PCR rate of 67%. Of course, there's increasing toxicity as you go up to this multi-experimental arm group. Nonetheless, though, suggesting there may be a benefit of bevacizumab and there may be a benefit of carboplatin in triple negatives. I show you that slide in this talk because at San Antonio, we presented data showing that these different immune cell signatures were also able to predict a higher likelihood of a pathologic complete response in patients receiving basically chemotherapy, right? So somehow having immune cells is even a good feature to predict whether you respond to chemotherapy. We don't yet know if this is going to be a predictor of response to immune checkpoint inhibitors. I think it will be, but that needs to be tested. So in the last bit of talk here, we'll show just a little bit of data on immune checkpoint inhibitors, where in this case, Rita Nanda and others presented at ASCO on pembrolizumab in triple-negative patients. So until maybe a year or two ago, I didn't know much about immune cells. Had to get out the textbook on immune cells and look up what some of these different proteins were doing. Basically, PD-1 and CTLA-4 are proteins expressed on T cells that, when activated, inhibit those T cells and prevent them from doing their effector functions. So that's why when we give inhibitors of PD-1 and inhibitors of CTLA-4, in essence, you're activating the immune system. And then it looks like the immune system has basically been kept in check, right? And now it can attack the tumor, right? So what Rita showed in this study with this waterfall plot, when given single-agent pembrolizumab to triple-negative patients, there were a good number of patients who had a response. And of course, as this audience appreciates, in triple-negative patients, there's a good number of patients who had no response, right? And so if this result holds up in larger studies, then there may be a role for immune checkpoint inhibitors in triple-negative patients. This also suggests there's going to be a role for biomarkers to predict this, because I don't think we would want to give this drug to everyone when it looks like about half the patients may not benefit, right? And so in this country, in my own country, we all know these drugs are very expensive, and I don't think we should just give them to everyone. We need to figure out what subset of patients benefit. That subset needs to be more specific than just triple-negative breast cancer patients. And so a great deal more work should be done to figure out if we can identify these patients versus those. So in closing, I hopefully have convinced you that basal-like breast cancer is the major constituent of triple-negative breast cancer. And if we're going to make therapeutic progress against triple negatives, we really need to target basals. And that means we should pay attention to the genetics of the basals, the driving genes of the basals, and potentially the immune epitopes that might be present in basal-like cancers. I didn't show you the data, but I can show you the publications, again from the TCGA, one from Cell of last year, showing that basal-likes have actually genetically very similar to ovarian cancers and very similar to lung squamous cancers. And actually, on a genetic level, these three are very similar, and actually all three are very different than ER-positive or HER2-positive breast cancers. Basal-like breast cancers, I think, reassuringly show many different drug targets, including the immune system, the PI3 kinase pathway, the RAS-MEK pathway, and potential angiogenesis. So I am optimistic that we will have the trastuzumab, or we will have an endocrine therapy type targeted agent for triple negatives or for basals, right? I've now been saying this for like more than 5 years now, but I think there's some promising leads on the horizon. Or not on the horizon, there's some promising leads in the room. And finally, actually, and this is best shown in what will be the talk I give tomorrow, right? Drug responsiveness is really dependent on multiple things. The biology of the tumor, the biology of the microenvironment, and the genetics of the tumor, right? And for predicting outcomes, we really need to look at all these things, right? So in the long run, we might have to run multiple biomarkers. We're already doing that, right? ER, PR, HER2. I think we just need to add a few more to the list. And in addition, not just adding them to the list, but adding them to a list in a biologically holistic and meaningful way, such that when we see you have this biology, it means we give these drugs for these reasons. So in closing, I'll just thank the many people involved. As you can all appreciate, this is a multidisciplinary project involving, of course, people in the laboratory, people in the clinic. Lisa Carey, Matt Ellis are my very close clinical collaborators and have taught me all I know about oncology. Pathologists, bioinformaticians, statisticians, and of course, the many cooperative groups and the funding sources for which, we need these to make advances in clinical care. So thank you for your time and attention.